So you're a home mycologist and you're starting to take mycology a bit more seriously. And so you've successfully cultivated a few interesting fungal strains, and congratulations for that. But now comes the hard part. How do you make sure that they're still around next year or even 5 years from now? How do you preserve these fungal specimens that you put so much effort into collecting and acquiring? Sometimes you have to isolate from the wild. Sometimes you get these strains from a friend, or sometimes you order them. But either way, you have to find a way to preserve them reliably. And so in this video, I'm going to show you how to solve that problem. I'm going to walk you through creating reliable agar slants and setting up glycerol stocks for cryopreservation of your fungal strains, so you can finally store your cultures with confidence. So how does this actually work? Well, in the case of glycerol stocks, we are essentially using a form of cryoprotection to conserve our fungal stock. When you freeze a living cell, the water inside it forms sharp ice crystals that act like tiny little knives, literally ripping the cell apart and killing it. Glycerol is what stops this. It's a type of alcohol that easily penetrates the fungal cell, and when it does, it replaces a large portion of the water inside the cell. This does two crucial things. First, it physically gets in the way of ice crystals forming, and second, it dramatically lowers the freezing point. And the result of that is, instead of a violent crystalline freeze, the cell enters a state of suspended animation, a glass-like vitrified state where it can safely hibernate for years, just waiting to be thawed and brought back to life. So that is a glycerol stock. An agar slant is a little different, and it's prepared by making agar and then pouring it into a test tube or a conical flask, and then letting that agar set inside of the flask at a 45-degree angle so that it maximizes the surface area available for your fungus to grow on. And that provides, of course, everything the culture needs to survive, but in a way that puts it into slow motion growth. When the fungus has the 45-degree angle plane of agar to grow on, and then you put it in a fridge at 4°C, what you do is dramatically slow the fungus's metabolism without completely halting it. So that said, let's then make a glycerol stock and also transfer a fungal sample from an old agar slant to a new one so you can visualize the process. First, again, prepare some agar, pour it into a tube, taking care to allow the agar to solidify at a 45-degree angle. And once it is properly set, take a tissue sample from an old slant, and in this case, this slant is 4 years old, very old for an agar slant, but I had faith it would grow, and lo and behold it did, on both a new agar slant and on 2 other petri dishes. So this goes to show you the longevity of these agar slants. This slant was created 4 years ago and kept at 4°C in my refrigerator, and also survived a move in which it was out of 4°C and at room temperature for a few days, and it still remained viable and grew. But again, at this stage, once you see the initial growth start from the agar slant, after keeping it at room temperature for a few days when you initially inoculate, or whatever temperature the fungus you're growing in this slant likes, once you see that initial growth, put it into 4°C, and then the fungus will grow very slowly from here. Now, one of the downsides with the fact that it is in fact growing is that anytime a cell divides, there's a small chance of a genetic mutation occurring during DNA replication, of course. So over many months or years, a slant culture will undergo a small number of cell divisions, and this means that the genetics of the culture you pull out of a slant a year or 2 or even a couple months later are not guaranteed to be 100% an identical clone of the single piece of mycelia you first put in. There is a potential for very minor genetic drift. Now, for the home mycologist, this isn't a huge concern, but of course for a research lab, it can result in the loss of virulence or kicking out a plasmid used to insert or remove a gene or anything like that. So yes, for an actual mycology research lab, this is a concern. And in this respect, glycerol stocks then are superior for genetic preservation in comparison to agar slants. The cryopreservation process halts metabolism almost completely, effectively freezing the genetics in time. So a culture revived from glycerol stock is a much more faithful genetic snapshot of the original culture. And with any process like this, yes, it's possible that you're gonna get some variation, but much less so with glycerol stocks. Given that, let's also make a glycerol stock. Now, the first thing you're gonna do is to get the materials. Acquire some glycerol. On Amazon, you can buy it as glycerin. Acquire some cryo vials or even some Eppendorf tubes. They work just as well. And those are also relatively cheap on Amazon. And you're gonna use a pressure cooker to sterilize your glycerol. And then you're going to dilute your sterile glycerol solution to 50% with sterile distilled water. And then from this half water, half glycerol solution, you're gonna fill up your cryo vial to about 2 milliliters. And then from whatever fungus that you want to take and create a glycerol stock from, you're going to take preferably a spore solution you made, but if it's not sporulating, if it's a yeast or if it's a basidiomycete that you have to bring to fruit in order to get it to sporulate, mycelia will often work just as well, but the standard is a spore solution. And you're gonna pour a drop or even 100 microliters into the cryo vial that has the 50% glycerol solution. You're gonna vortex that, you're gonna label it, just so you know what it is, and you're gonna write the date on there, and then just put it in the freezer, and that's it. Now, in an actual mycological research lab, they're going to have a -80 C freezer to store your glycerol stock in, and at this temperature, fungal stocks can easily remain viable for decades. In the case of your at-home freezer, obviously you don't have the luxury of a -80. The temperature in your freezer is gonna hover around -18 C. And under these circumstances, yes, your fungal stocks will tend to fail at a much higher rate. But that said, there have been studies done out there that measured glycerol stock viability at these temperatures, and they do in fact survive, some of them for years at this -18 C temperature. For my at-home fungal library, I reactivate from glycerol stock yearly and have not had any fail. And in order to reactivate, after you make your glycerol stock and you have it in a freezer, now you want to make a plate of whatever fungus that you're gonna use, and you wanna test it for viability. Wait for a couple weeks after you initially make the glycerol stock, and then basically get yourself a plate, some sterile toothpicks, dip the sterile toothpick into the glycerol stock, and this should then contain some of the cryopreserved cells of that fungus. And I like to then streak this toothpick on the plate in a tic-tac-toe pattern. Following this, simply incubate your inoculated plate at the temperature it grows in. And if you see fungal growth, you know your glycerol stock is viable. So then, folks, that is how you preserve your fungal strains, your fungal collection, your fungal library for years to come, so that you don't have to buy plates or liquid culture of these fungal isolates that you've worked so hard to acquire. So again, I'm trying to bring to you guys home mycology techniques, because we all don't have the luxury of a, fully stocked, fully equipped fungal research lab, but I'm trying to democratize fungal study, because it shouldn't be gatekept by these universities. And so therefore, if you like this channel, if you like the material, please go ahead and subscribe. If you're watching this on TikTok, subscribe to my YouTube. If you're watching this on YouTube, subscribe to my TikTok, and I will bring you guys much more fungal content to come. Thanks for watching.